Welcome back. In the previous lecture, we discussed the primary treatment of CVS treatment. Now in this lecture, we'll talk about secondary treatment. Okay. What do we do in secondary treatment? Now we know that from primary treatment, we are bringing that all that liquid which is called as primary effluent, right? So in primary treatment, what did we do? First we have done filtration and we removed all that floating debris and all, and then we brought it to a sedimentation. Now all the sediments are being removed off. Only the liquid is collected, right? Only this liquid is collected. This liquid is now brought to secondary treatment. The liquid which you are collecting is called as primary effluent. So we can start secondary treatment with primary effluent. The primary effluent is now sent into this primary effluent liquid is now sent into aeration tanks. Now, how does an aeration tank look like? From here, we should take it into the aeration tank this way. From here, it is connected to, or else, shall we draw it here itself? We'll write the matter later. We'll continue the diagram here itself. Now, this is now sent into an aeration tank. This is an aeration tank. All the liquid from the primary treatment has collected here. Now, since the name itself is aeration tank, right? So, we need to continuously supply oxygen. So, we need to aerate it. Oxygen, 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 oxygen. If the oxygen has to dissolve, if it has to mix, we need to have an agitator system also. Take a rod-like structure and then rotate it. So what is this called? Agitator. This agitator mixes the contents. Now what is there? Primary effluent is there. What is there in the primary effluent? Heterotrophic bacteria are there. Fungi are there. Food is already there. Now in this primary sludge, what is there in this primary effluent? The primary effluent means this liquid after primary treatment. What does it have? It has heterotrophic organisms. It has heterotrophic microbes plus organic materials, organic matter also it has. And you are providing optimal conditions. You are doing aeration here. And you are doing agitation. When you are providing air, when you are agitating the heterotrophic microorganisms which are present here, they start degrading the organic matter. They start degrading the organic matter, and with that organic matter they are nicely growing. So the bacteria and fungi form flocks. They form flocks. The fungi, we know it is a mycelium. It will make a mycelial network. The fungi will make mycelial network, and between these mycelial network, bacterial cells will be trapped off. This complete structure is called as flock. The bacteria are trapped within the fungal filaments. Fungal mycelium is called as flock. This flock, it is exposed to aeration, exposed to oxidation, and it can decompose the organic matter. This mat-like structure, what it will do? In the presence of oxygen, it starts degrading the organic matter. Now for this particular primary effluent, if you see, what is the BOD? Since it is sewage, it has high BOD. Since it is sewage, the primary effluent, it has high BOD. What do you mean by BOD? We need to understand here. BOD stands for biological oxygen demand. It stands for biological oxygen demand. Now, what do you mean by biological oxygen demand? Now, if the organic matter is more here, then to break the organic matter, the bacteria will grow more. If they are growing more, they demand more oxygen, right? If it is freshwater, organic matter will not be there. If organic matter is not there, bacteria will not grow. If bacteria are not growing, they are not consuming the oxygen. So that means fresh water will not have BOD, least BOD it will have. Sewage water, more the organic matter, more the bacteria will grow, more the oxygen they are consuming. So what is BOD? You understood that BOD refers to the amount of oxygen consumed. The amount of oxygen consumed by the heterotrophic bacteria to eat off the organic matter. If you want to give the definition, we can tell BOD refers to the amount of oxygen consumed by all the organic matter present in 1 liter of the sample to oxidize the organic matter. Let us write down the definition. BOD, it refers to the amount of oxygen consumed, the amount of oxygen consumed by oxygen consumed to remove or to oxidize the organic matter present in 1 liter of sample. In 1 liter of sample, this is 1 liter of sample. In 1 liter of sample, some organic matter is there. To remove that organic matter in this 1 liter of sample, the bacteria will consume how many milligrams of oxygen is the demand. It's BOD. Now is it clear? Think this is 1 liter of sewage. To remove the organic matter present in this 1 liter of sewage, now the amount of oxygen in milligrams consumed by the bacteria to remove the organic matter is called as BOD. So that means BOD is a measure. What is BOD? It is a measure of rate of uptake of oxygen by the microorganisms, correct? So we can tell that BOD is the measure of rate of uptake of oxygen by the microbes. What is BOD, Amma? BOD is a measure of oxygen uptake by the microorganisms. Where are the microorganisms present? Present in the sample water. By the microbes, where are the microorganisms present? Present in the sample water. So, if the organic matter is more here, then bacteria will grow more. If bacteria are growing more, they require oxygen more. So, high organic matter, higher the organic matter, then we will tell high is the BOD. High is the BOD means high the organic matter means high the pollution. So means it is highly polluted water. It is highly polluted water. Understood? So, BOD, are you measuring the organic matter directly or you are measuring the oxygen consumed by the bacteria? Come on, tell me. We are measuring the oxygen consumed by the bacteria. So, then BOD, is it a direct parameter or it is indirect? We tell it is an indirect parameter since we are not checking the organic matter directly. We are checking the oxygen consumed by the bacteria. Thus, indirectly BOD is a measure, right, of organic matter present in the water. Can I write it here? Thus, indirectly BOD is the measure. It is the measure of organic matter. We tell that BOD is indirectly helping us to find out the organic matter, where BOD will directly help us to find out the oxygen consumed, right? So BOD indirectly is a measure of organic matter present in the water. Organic matter present in the water. Children, this is very important point, and this point is there in NEET exemplar also. BOD, is it a direct parameter or indirect parameter to check the organic matter? It is an indirect parameter, right? What is direct parameter? Oxygen consumption is a direct parameter, whereas organic material degradation, it comes as an indirect parameter. What are we checking here? How much oxygen is being consumed. Now, if it consumes 1500 milligrams of oxygen per liter, then such a type of sewage, we'll call it as weak sewage, or it's a weak waste. Okay. And if it consumes 1500 to 1600, then we will call it moderate waste. If it consumes this much amount of oxygen, then we call it as moderate. I am giving the BOD values, right? And if it consumes more than 1500, more than 1600 milligrams of oxygen, if it is consuming, it's a strong waste, it is a highly polluted water. If it consumes, if the BOD value is more than 1600 mg per liter, then call it as heavily polluted sewage. It is highly polluted waste. It is highly polluted waste. I tried to explain what happens in the aeration tank, which is the first step under secondary treatment. This is the first step under primary treatment. This is the second step. Under secondary treatment, this is the first one, but in order, it is third one. In aeration tank, you are bringing the primary effluent, the liquid. The liquid is dropped into the aeration tank. You will supply air. You will mix it with the help of this agitation. You will do. What is there here? Sewage is there with the organic matter, with the microorganisms, bacteria and fungi. They nicely eat off the organic matter when you supply oxygen. The more the oxygen consumption, the more is the organic matter. The oxygen consumption means oxygen demand. We measure it in terms of BOD. BOD definition is the amount of oxygen consumed to remove the organic matter present in 1 liter of sample. If you want the exact definition, at 20 degrees centigrade also you can add. At 20 degrees centigrade temperature also we can add. Since BOD is measuring the oxygen consumption, not the organic matter decomposition, it becomes an indirect parameter. These are the BOD values. If it is more than 1600 milliliters, that means the sewage sample is highly polluted. So we are going to continue the process in aeration tank till the BOD drops down. So we'll continue aeration and agitation till the BOD levels, go less than, till they go less than 10 to 15% of the raw sewage. Till the BOD levels reduce to 10 to 15%, do aeration, agitation, the bacteria will grow nicely, they will eat off the organic matter. When the BOD drops down significantly to 10 to 15%, now it's a time to take from aeration tank to the next level. Take a screenshot, then I will continue. Yes. Now we can tell after the BOD significantly drops down, so we need to collect this and now take it to a settling tank. Whatever the liquid is there, in which organic matter is not there. This liquid is called as, what is this liquid called as? The liquid is called as effluent. This effluent which came from here is primary effluent. Now this effluent, you can call it as secondary effluent. The secondary effluent is somewhat clear now. The secondary effluent can be released into the water bodies. The secondary effluent can be released. It can be released into water bodies. The secondary effluent, this liquid will be released into the water bodies. Whereas whatever the solid which is made here is called as activated sludge. So as a result of this, whatever the solid is formed here. So this solid, what is this solid called? The solid which is left out, you collected all the liquid. That liquid is called as secondary effluent, and this is trapped within the filaments, right? Fungal filaments. Now this one, what is this called? This is called activated sludge. This is called activated sludge. Amma, from here take all the liquid and release into the water bodies. Take all the solids, bring it to the step 4, which is called as settling tank. After removing everything, will this container become vacant? Yes. Can I erase the things which are present here? See, all this gets removed because from here we took the water liquid and we sent it into the water bodies. The solids are dropped here. Means it is absolutely free now. This is absolutely free now. Now it has an agitator, right? It has an agitator. Now in this settling tank, now we will allow the sludge to settle. The activated sludge is settled down. It is settled down in settling tank. It is settled down in settling tank. Now from here again the fresh sewage will come, right? To this fresh sewage again bacteria and fungi need to grow and they need to reduce the BOD. So where are the bacteria and fungi? Here are the bacteria and fungi. So what we do when you want to make a curd, yesterday's curd sample little you will add to the milk. In the same manner, take little of the activated sludge from here and drop it here. Take little of activated sludge and drop it here. This sludge, like the yesterday's curd sample, it acts as an inoculum for the fresh sewage. Now what is this called? This is called inoculum. So this activated sludge, little is dropped into the aeration tank back to act as an inoculum for the second stage. Now from here again fresh water will come, right? Sewage water. For this sewage, this acts as an activated sludge. It acts as an inoculum and it will start the process fast. Now let us see what is happening here. Nicely they are settling. Here they worked rigorously to remove the organic matter. They have given some time to rest it now. Now in settling tank they are resting. Now from the settling tank, now we will now pass it on to another tank. We will now pass it on to another tank. This tank is called as anaerobic sludge digester. So all these I kept open because aerobic process is going on. There it is, anaerobic sludge digester. So completely seal it. Completely close it. It's an anaerobic sludge digester. So all that settled matter, the activated sludge, is now pumped into the anaerobic sludge digester. All together I am taking it here now. All this is now taken into anaerobic sludge digester. The activated sludge is taken into the anaerobic sludge digester. Now, in absence of oxygen, in the anaerobic sludge digester, in absence of oxygen, let me write down. In absence of oxygen, what happens to that normal bacteria and fungi which needs oxygen? They will die. In absence of oxygen, bacteria and fungi will die. They will die, and another set of organisms will flourish now under anaerobic conditions, and another set of organisms will now grow. You are not adding, they are there only, will now grow under anaerobic conditions. Under anaerobic conditions. Amma, what do you mean by anaerobic conditions? Absence of oxygen is called anaerobic conditions. What are those organisms which will grow? So they are two types. One are called as non-methanogenic bacteria. Non-methanogenic bacteria is one set of bacteria. The other set of bacteria, the main set of bacteria, are methanogenic bacteria. The main set of bacteria are methanogenic bacteria, will flourish in anaerobic conditions. They will flourish in anaerobic conditions. And what are they going to do now? Whatever left out matter is there, whatever organic acids and all are there, these methanogenic bacteria, methanobacterium, methanococcus and all, they will flourish in anaerobic conditions, and they convert the remaining organic acids they convert them into marsh gas, gobar gas, hydrogen sulfide, carbon dioxide. So what are they doing? They are just converting under anaerobic conditions whatever the 10 to 15% BOD and organic matter is there. They are converting that organic matter, organic acids, into biogas or marsh gas, which is methane. Then H2S and carbon dioxide are formed. So anaerobic sludge digestion is the last step of C-waste treatment. Understood, children? In this session, we started with secondary treatment. Secondary treatment, what is your starting material? Primary effluent. So, we will collect the primary effluent. From here, we will bring it to aeration tank. We provide air, we provide agitation, we mix it nicely. The heterotrophic bacteria consume air and they will mix nicely. They eat of the organic matter. While they are eating of the organic matter, they consume the oxygen what you have given. They create a demand which is called BOD. Polluted water, more organic matter, more bacteria growth, high BOD. So you will be checking the BOD and you will do the aeration tank till the BOD drops down very less, 10 to 15%. Till then you do here aeration tank. In this aeration tank, bacteria are nicely eating off the food. They grow like filaments, they grow like flocks. The flocks contains bacteria wrapped by the fungal filaments. After the BOD value goes down, take entire thing and take it to the next tank, which is called a settling tank. In that settling tank, we will take only the solid part, only the flocks we will take. Whereas the liquid, you can directly dispose into the water bodies because organic matter is being removed off. Now this settling tank, whatever the solid is there, is called activated sludge. Allow it to cool down. Now it will nicely settle. Now in that, take little bit of sludge and drop it back in the aeration tank, which acts as an inoculum for the fresh batch. Which acts as an inoculum for the fresh batch which is coming through this way. Now then take the settling tank, take all these things into a next level called anaerobic sludge digester. Till now, 1, 2, 3, 4, all these are aerobic processes. Now when you provide anaerobic conditions, oxygen absent conditions, then this bacteria and fungi which are there normally will die because they are aerobic. Now another group of bacteria which are called anaerobic bacteria will grow now. They belong to two categories: non-methanogenic bacteria, methanogenic bacteria. Methanogenic bacteria will flourish more, will grow more, and they consume the leftover organic matter and they break them into methane, which is called marsh gas, biogas, carbon dioxide and hydrogen sulfide gases. So completely the solid is also broken down, the liquid is already released into the water bodies. So this is called as secondary treatment. Take a screenshot. Right, so in NCERT the information is given till here only, but usually we see that there is tertiary treatment also. So in primary treatment it is physical treatment, in secondary treatment it is biological treatment. If we have to list down tertiary treatment, it is chemical treatment, where we try to add chlorine and all, disinfectants and all before releasing that water into the water bodies. So usually in NCERT tertiary treatment is not there, but for your understanding, so we can include tertiary treatment also. Tertiary treatment, it comes under chemical treatment. So in chemical treatment, what do we do? The secondary effluent, the fluid which is there, the secondary effluent is treated with chlorine. The secondary effluent, the liquid which is coming from the aeration tank, is treated with chlorine and other disinfectants okay, before releasing into the water bodies. Before you release that water into water bodies, we need to add chlorine, we need to add other disinfectants. This comes under tertiary treatment. Okay children, with this we finish sewage treatment. Since if you don't treat the sewage, it causes many problems, the government of India has initiated Ganga Action Plan, Yamuna Action Plan to purify our sacred rivers, to purify our water bodies. Now, how do they do it? If it is a small tank, you will clean it, you will dry it, and then you will add fresh water. This is a river which is flowing from that state, passing these many states and going there, right? So how will you clean such a big river? Now this Ganga Action Plan, it is not a small thing, right? So first what we have to do, so we have to at a particular area, we need to take all the water and we need to do all these steps, primary treatment, secondary treatment, and reduce its BOD and release the water again. So after 2 kilometers in the same river, again here, take again the water, again process it, send it back. Again after 2 kilometers, take it out, process it, send it back. So you are doing this, and this water will not be there here, right? It comes here. This comes here. It mixes up with the old water. So you have to do it continuously for a period of time, and we need to check the BOD all the time. So the water or the river is said to be pure if the BOD levels are brought down. So by doing this, we are protecting our environment also, and we are protecting our natural resources also, and in turn we are protecting ourselves from diseases also, waterborne diseases also. So the government of India has initiated and it has taken a plan like protecting our sacred rivers. Under that, Ganga Action Plan will come, Yamuna Action Plan will come. So under that, what do they do? In the flowing river, at one particular point, at a distance of 2 meters or 3 meters, 3 kilometers, they'll take the water from the river, they'll do the processing, and then they'll drop it back. Again, they'll take the water, they'll do processing, they'll drop it back. So they will do like this sequentially till the BOD comes down, and then the water is treated as a pure water. After that, they will not allow any waste to be decomposed into it. Okay children, hope the content is informative. We have discussed sewage treatment in detail. In the next lecture, we'll talk about biogas production. If you like the content, like, share, subscribe to my channel. Thank you.